Good morning, my dear students. I welcome you all for the bioscience class. In this class, we are going to discuss about the final topic of our lesson, that is photosynthesis versus respiration. Along with that, the activities that are pent over, that is increasing of temperature during the process of respiration. Let's look at the activity 3. In this activity, we are going to discuss how the lime water will be converted into milky white. So, to conduct this activity, we need to take a handful of Bengal grams seeds and soak in the water before you conduct the experiment. Keep the soaked seeds in a cloth pouch and tightly and string it tightly. Keep the cloth pouch in the corner of your classroom. Next day, collect the sprouts that are being germinated from the pouch. Keep in the glass bottle or a plastic bottle. Take a small beaker, fill with the 3 by 4th of lime water. So what we are going to be doing here, to conduct this activity, the most important material that are required are Bengal grams. The Bengal grams need to be sprouted. So to make the seeds to be sprouted one, we need to soak in the water before you conduct the experiment, and we need to collect this material. So after that, we need to take a small beaker with the lime water. Keep the small beaker in the large beaker carefully. Close the plastic bottle tightly. Make a similar set of unsprouted seeds. Keep these two sets undisturbed for 1 or 2 days. During this time, observe the color of lime water in the bottle set. In which set does the color is going to be changes. Your same type of identical sets should be prepared, but whatever the second set that you are going to be prepared must be not a sprouted one. Unsprouted seeds must be selected. So same process, two bigger huge bottles and place it in a corner and try to observe without disturbing for 1 day. What you are able to observe, you can see that the changes are going to be occurs in the lime water because of respiration process. Here is the activity 4. Take a sprouted seed that you have prepared in the activity. Along with that, we need to take a thermal flask. Remove the lid and prepare a cork, which must be have a small holes. Take care of your bulb of a thermometer should be dipped into the sprout. Close the flask with the tight bottle. Record the temperature every 2 hours. 2 hours. Advise them to conduct for 24 hours. In this activity 4, we are going to be using the thermal flask. Along with the thermal flask, we are going to be using the thermometer. In the activity 3, we have used the lime water. In the activity 4, we are going to be using the thermometer. So see that whenever you are going to be placing the thermometer into the thermal flask, try to see that the thermometer must be inserted or must be merged into the sprouted seed. So sir, where should I get the sprouted seed? From the activity 3 itself. Whatever the seeds that you have prepared in the activity 2, activity 3, the same seeds can be used for this activity 4 also. Here, taking the thermo flask and filling with the sprouted seeds and placing the thermometer and the lid must be closed with the cork. When you observe this one, the temperature will be rises. What is the reason why the temperature is going to be rises? Because it is going to be removing the lime, or it is going to be removing the carbon dioxide, and this carbon dioxide make the temperature to move forward. Here is the next final one, that is photosynthesis versus respiration. What is the major difference between the photosynthesis versus respiration? Plant carry out photosynthesis, which means that they produce their own food material from the atmosphere, carbon dioxide, using sunlight energy from the sun. Already we came to know that the plants are capable of preparing their own food material depending on the carbon dioxide that are being obtained from the atmosphere and the sunlight energy which is going to be getting from the sun. This process is a complex series and steps involved in the conversion of light energy into chemical energy, which is then used of synthesizing of a sugar and carbohydrate. So this process, the process of photosynthesis, is a very complex one. In this process, you can say that the complex substance will be converted into simple substance, and this modification lead to the synthesizing of a sugars and carbon dioxide. This process of synthesis is an anaerobic process which is going to be occurs in the chloroplast. It is an anaerobic process which is going to be occurs in the chloroplast. Just look at the equation given below for the summarization. 6CO2 plus 12H2O in presence of light and chlorophyll, it gives rise to C6H12O6 plus 6H2O plus 6O2. This is the equation of photosynthesis. Once the product is going to be formed in the form of sugar, can be used the process of respiration to provide the energy. Here the production of food is one step, but breaking down this food material is very important. The food that has been produced in the form of a sugar will be highly helps in a production of energy. Respiration, as we know, it is not just exchanging of gases, it is a process of breakdowning the complex substance food material or a catabolic process to produce the chemical potential energy. So respiration, in the lower classes you might have came to know that respiration is nothing but a just exchanging of is not just a production of energy. It is not just a taking a transfer of gases, but it is going to be helping for the production of potential energy. This can be summarized with the equation as, just look at the equation, 6 C6H12O6 plus 6O2 gives rise to 6CO2 plus 6H2O and energy. By this one, it is clear that glucose molecules are going to be broken down and produce the